Greetings everyone, my name is Alex, I'm a PhD in AI and CEO of Blackthorn.ai. I presented papers at NeurIPS and published in Nature, except academic research. For more than 25 years, we worked with companies like Bayer and Google, transforming advanced research to hands-on applied technologies and results. And we will talk about one of such advanced technologies today, and which is more beneficial for the auditory, how to turn it into practice for you. So stay tuned. I will share my screen now. And we will talk about protein engineering today. This is one of the most complicated, but also one of the most promising technologies in the modern AI plus biology world. Historically, protein engineering made a long way to what it looks like today. It started from directed evolution, where a library of random mutations was introduced, where bacteria got introduced vectors with random mutations of DNA that turned into plasmids and then got inserted into their circular DNAs. And then people, through directed evolution, tried to get the proteins they wanted. In parallel with that, another set of methods were developing. They called rational design, and those were about modifications, but guided by structure. So it's a set of force fields and different simulations, like Amber, Charm, and other things, where people had the structure and had some idea about the desired and predicted function. Around 2010, a new set of methods around deep learning networks emerged, and then around 2020 it evolved to a set of AI methods that allowed far more advanced capabilities compared to the previous ones. For example, desired mutations, proteins with desired functions, building structure from sequence, and enabling a de novo protein design. We will talk about this set of methods today, and before we will briefly talk about the problems of classical methods. So, major problem of classical methods is that they are time-consuming and expensive. This is what people want to cut in early drug discovery and drug development consequent. So development of one molecule now costs over $2 billion. It takes 12 to 18 months, and still 90% of candidates fail along clinical pipeline. In particular, protein engineering has issues like fixed topologies design. I mean, now we're talking about classical technologies, directed evolution, and structural design. So this talks about that we have a set of predefined topologies, right? We have some PDBs with coordinates, and it is very hard to go beyond that. And usually new functions, they are associated with new folds, new helixes, and old topologies just keep us away from that. So it's a limitation from topology. We rely just on existing structural data. If we are going to design some novel structures, that will be extremely hard. Also, the libraries, they are large, but they are very noisy. Different groups of scientists, different devices, different techniques, different accuracies, also mutations. It is impossible to cover all set of mutations. So usually it causes bias in our crystal structures of proteins, which further limits and biases anything that is built on top of that. Other set of challenges are practical. It's random destabilization. For example, by introducing one random mutation, and this is the essence of directed evolution. It's inserting a set of random mutations and seeing which phenotype works as expected and mutating it further, trying to get a better representation of a phenotype desired. So, but suddenly some mutations, they just ruin the structure of a protein and of course its function, because it's a function is the consequence of a structure. Also, biases of experimental methodologies. It's difficult to find sensitive and efficient method for screening because all screening methods, they record not actual structure, but the screen structure. Yes, screening methods itself, they introduce error and bias, like PLR and other things. So the other wing is time-consuming and expensive. All the methodologies, like before AI, we're talking the before AI age now, they are based on the trial and error nature. What it means? It means we introduce as many mutations in a genotype of bacteria as possible in the structural gene, and then we're hoping to find the one that works. And this cycle is very long, right? So we need to synthesize those peptides, we need to inject them, we need to inject vectors for transferring the gene into the bacteria. Then we need to get this protein, then we need to measure its function, and it's a difficult and time-consuming way. So there is no fast way and there is no general way. Every story is unique. Also, most of the time, it's just brute forcing experimental library screening. Also, during all of this process, we are not learning on our mistakes. Every new set of mutagenesis may introduce and definitely introduces past errors. There is no way to deal with that. It is just how it works. We cannot limit it. So there will be a lot of failed attempts. And of course, people who do it will pay for these attempts. So in this lecture, we want to somehow deal with this, and AI is the right tool to help in these circumstances. So briefly, we talked about directed evolution, right? We have a genome, we introduce mutations, we have different phenotypes. Through random mutagenesis, we have like different recombinations through a single nucleotide, polymorphism, insertions, deletions, and replacement and other things. This is directed evolution, and rational design actually starts from a structure, and then we go from a structure to a genome, and then we synthesize from a computational structure. I apologize. We have a simulated structure which we expect to have some function, and then we do a mutation, then we check it. And there is an intersection of these methods is a semi-rational design. It is where we perform mutagenesis not for the whole protein, but for some desired part of it which needs improvement. For example, typically it's binding site. Right, so in 2010 emerged deep learning, as well as other industries. It affected life sciences and drug discovery. In 2010, it was a set of separate models, each of them predicting some small property, or one of them predicted affinity, another one predicted coordinates. So it was piece by piece. Then closer to 2020, all of these small models with growth of computational capabilities and data, they emerged into larger and conceptually different, conceptually the same, but much more scalable approach of applying AI-based generative models. And this gave us a whole new set of opportunities about which we will talk in this webinar. Briefly about data growth. So around 1917, 2000s, and early databases emerged such as PDB and UniProt, and there were like around 100 proteins in each of them, totaling to like around 10,000 sequences. but they grew like 100,000 and like thousands times. Now they are like tens of thousands of structures and millions of sequences. In 2010, high throughput screening was invented, and that led to an explosion in genomic and proteomic data. Now we have hundreds of millions of sequences, and now, except of experimental like crystal structures, we have simulated structures. In 2020 was AlphaFold introduced, and it allowed to simulate a lot of structures. So now we have hundreds of millions of structures, and part of them experimental, and part of them is simulated for unknown proteins. So all of this, like growth of computational resources plus growth of data availability, allowed to create a new generation of models, which are actually now we call AI-guided protein engineering. Nowadays, the benchmark, these systems can save up to 60% in costs, and making the discovery of proteins 2 to 5 times faster. I will show you examples like just soon, and overcome limitations like avoid this random mutagenases, avoid an infinite loop of trial and error, expensive and time consuming. And actually now protein engineering is folded into 3 groups. First are structure-based methods. It is where the structure is more or less known, but we want to optimize some pockets or make the structure more stable or able for oral way of administration and other things. In this way, we play a little bit with the amino acids, and then we fold it back, but we know the scaffold. So we're playing around the scaffold. Also, this set of methods oftentimes combined with the molecular dynamics, so we can evaluate the energy and find the most stable structure. So AI kind of proposes different amino acids, folds them, and now like MD computes the energy. Then we select the most potentially stable complex. We can even simulate it with water and other solvents. So fold number B are a set of sequence-based models. Sequence-based models means that the input is not a structure. Here we have PDB, 3D structure with coordinates of some core amino acids or atoms. And here we have just a sequence of amino acids. Like in case of FASTA, it's a FASTA letters, and MSA is a multi-sequence alignment. We have stars, dots, and double dots, which stand for different replacements, equivalence, sense, and missense replacements. So this, it's a primary protein structure, then is fed into some kind of AI model. Typically, it's a transformers nowadays. Yeah, fancy names is LLM, and protein and language of amino acids is ultimately a language. So yeah, LLMs can talk protein, and then we can suggest mutations for desired function, or we can fold this primary structure into secondary, tertiary, and like into a structure, a 3D structure. The third fold of methods is called de novo design. De novo design is when we want to design a completely new protein structure with desired function. Yeah, so kind of we want a drug to treat diabetes, for example. Of course, diabetes is indication, right? But the function could be inhibit that kind of a receptor, like GLP-1 receptor. And then, based on a whole set of knowledge and rules of folding of proteins, we feed the model with some random noise or scaffold. Yeah, depending, are we want to get a completely new structure, or are we still want to stay around some scaffold? and build around, and we get an output, expected output. Moreover, we can check its affinity to desired target with further methods, right? So the whole idea is that now we skip this circle of samsara, of suffering, of trying different mutations without knowing which one leads to the desired outcome, and save a lot of money and time on wet lab experimentation and also like theoretical construction. So let's talk about money and why it is important now. First of all, market, even all the biotech now is in decline. Protein engineering market is growing. And it is predicted to grow further. As you see, it grows slightly exponentially. So there will be more and more, like it's expanding exponentially, more and more demand in the future. Venture funding for protein startups are just insane. I heard about cases around like 400 million, like something insane for, to be honest, the quite simple technologies. And also, in addition to that, major pharma companies, they also rely on external innovation, like partnerships, acquisitions. So they buy these startups, they work with external partners to develop some new technologies and apply on practice. I know about examples from Bayer and Sanofi. Also, it has been proven that the clinical level of acceptance of therapeutic proteins is higher than small molecules, right? Maybe because the field is new and there are just more new potentially therapeutic proteins than small molecules, because the small molecules field is like comparatively old to protein. I'm not saying that it is bad or it is inefficient. Yeah, there is still a lot of place for small molecules, but probably there are just more unknown proteins rather than small molecules in the world. But the space of both are just insane. Then, from examples known, we see that it reduces the drug discovery pipeline 2 to 5 times in time and cost. And yeah, also covers half of the costs. Now let's talk about a couple of successes of AI-powered drug discovery. Probably you heard about rentozatib. It is the first AI-designed drug which entered phase 2 clinical trials developed by in silico medicine. It reached preclinical development only in 18 months and advanced to phase 1 of clinical human trials in just under 30 months, which is fast. Usually this phase is usually 2 to 5 years, something like that. In this case, we see it was 2 years, so fast, but not lightning fast. So around 2 to 3 times. Early safety therapeutic potential was confirmed. Yeah, you know its outcome of phase 1. Another case is GLP-1 set of peptides. Here the goal, again it's in silico, combine its platform Biology 42 and they run completely novel protein design, generated over 5,000 GLP-1 receptor peptides in 72 hours, as they claim, without using known binders. So they went completely blind and it worked. 14 of them showed activity and 3 of them were strong binders showing nanomolecular potency. Here you can see the examples. Another case is halicin, is AI discovered antibiotic against drug-resistant bacteria. It was a collaboration between MIT and a pharma company, and they developed a novel antibiotic effective agent against drug-resistant bacteria. You can see here it on the screen. So their AI system screened over 100 million of compounds in just 3 days and found 23 promising candidates, with 8 showing antibacterial activity and 2 demonstrated strong potency. So for 3 days, that's quite nice. More about collaboration. Biorecursion just signed a deal, 1.5 billion AI aliens that will unite a real world, which Bayer can give, and a world of simulations and AI, which is under recursion control. So they will learn based on the real world evidence and build more hypotheses. Also, a part of a deal is NVIDIA by a Hive 1 supercomputer, and their goal is to screen 1.7 million small molecules across 50 cell types to identify new therapeutics. To summarize, we see from evidence that AI design has real results. There are very big collaborations, like large money involved. And by fact, AI design compounds speed up the drug discovery pipeline 2 to 3 times compared to traditional approaches, of course. So how typically it works? Here is the AI-powered protein engineering pipeline, and there are a number of typical steps that you usually take. First of all, you need to collect data, train model, run predictions, try them in the lab, see how it works, extend your data set, maybe remove some outliers, and actually do it again. And in maybe if you are good and lucky, right, and not doing it first time, it will work from the first time, so you will not need feedback loop. But most of the time you need to do some additional work, experimental validation, like IC50, and other things, tuning. Okay, now let's talk about actually models, the most modern, the newest models that are available in the field. And there are 3 top players here. The first one is AlphaFold 3, second Boltz 2, and the third is OpenFold 3. Let's talk about their key features. So here you can see an example of a folded structure with the AlphaFold. Yeah, we folded it. So there is a ground truth in gray and folded structure by AlphaFold in green and blue, right? You can see, yeah, there is some quite a discrepancy, but not significant, and the largest are on loose ends. The ground structure is a crystal structure, and here you can see its uniprot number. AlphaFold, yeah, we will talk about the specificity and yeah, drawbacks of AlphaFold, that it is proprietary, so it's a closed model, and companies oftentimes stay away from using it, yeah, because they are afraid that it will learn on their data. Maybe yes, maybe no, who knows? Probably yes, rather than no. But there are strong benefits of AlphaFold. Yeah, first, it was a first player in the field. Second, all other players are built on top of its architecture. Third, it can work with different modalities like DNA, RNA, proteins, oligos, and other things. But it has more restrictive license and less flexible functional capabilities. So if you want to play with the model, fine-tune it, tweak hyperparameters, they will not allow it. Just use API. Also, it is known that AlphaFold 3 was trained on the 300 million structures, but the dataset is unavailable, yeah, which is another drawback, yeah, because if you want to somehow validate it, you need new structures that are not present in original dataset. Otherwise, they will just be biased and overfitted, very good results. So it is hard to know what structures were not there. Nonetheless, it is known that AlphaFold 3 was trained on structures which is younger than 2023. And if you take any structure that is past that date, a novel, probably that it will not be in the dataset, and it's okay case for validation. So Boltz 2, Actually, it's a fork of AlphaFold, but with a new heads. As you know, LLM has multiple attention heads, and Boltz 2, except of structural head and energy head, has affinity head. Trained on around 80,000 curated results and affinities like IC50, Ki, inhibition coefficient, dissociation coefficient. Boltz 2 knows it's mostly made for affinity studies like binders. Another big benefit is that it is open source model. You can tune it. It can model local molecular dynamics, right, and achieves good accuracy. From our experience, Boltz 2 is over optimistic. So it finds more pairs than anybody else, and IPTMs for them are quite high, while other methods, they are not so positive. Yeah, so keep this in mind. Finally, OpenFold 3. So OpenFold 3 is just an open source fork of AlphaFold 3. OpenFold 3 uses for computations instead of floats. In AlphaFold, it uses bits. That's why the model is much more lightweight, right? But it is hard to expect from it something more than from AlphaFold 3. By the way, it is something to discuss because simpler model means that it overfits less, right? How to differentiate? Case by case. So typically OpenFold, since it has an open source license, they are freely used by corporations and consultants, small, large, mid-sized companies for drug discovery. It was released just recently. Yeah, it is hard, but we tested it, and here is the one of the complexes. Again, the gray structure is the original one, and the blue one is folded. By the way, here you have ligand, right? So it's a complex. It's a complex of. By the way, OpenFold 3, in the same way as AlphaFold 3, works with proteins, small molecules, DNA, RNA, and peptides. Right? We can see examples here. Of course, our interest didn't stop here, and we tried to went further. And here is a comparison table. AlphaFold can simulate structure of proteins, run post-translational modification, and usually outputs high accuracy results, especially for large complexes and multimeric assemblies. AlphaFold second multimer became a gold standard for protein-protein interaction. As opposed to that, Boltz 2 works mostly with protein and protein complexes, can output binding affinity. Yeah, this is something AlphaFold doesn't do, and approaches high accuracy and works fast. OpenFold is same as AlphaFold 3, but yeah, more lightweight. AlphaFold 3 dataset is unavailable. Boltz 2 dataset experimental, so it's the same architecture, but they trained it on a different dataset. It's all PDBs released before 2023, and for binding affinity, 18,000 systems, like I said, IC50, Ki, Kd, and some distillation data for different modalities. OpenFold 3 trained on the 300,000 experimental molecular structures from PDB, UniProt, and others. More than 40 million synthetic structures, right? So it's a mix of real world and simulated, and also more than 20,000 protein drug pairs. Another crucial difference probably is that Boltz 2 and OpenFold implemented in just PyTorch. So yeah, it's easy to work with. Any ML engineer knows TensorFlow and PyTorch. And it's easily scalable across multiple GPUs. It's adopted in different corporations and companies and stuff. And AlphaFold is written in JAX. JAX is a library of Python for high performance computation, for GPUs and stuff. It is more sophisticated, but beneficial for large scale tensor operations. Less flexible, more difficult for inexperienced users. However, it is more performant. License, right? AlphaFold 3 Creative Commons, and here is permissive license. You can use this for your own products and commercial purposes without any fear. Okay. Now, even more, we found 10 oligos. Oligos mean that we had two branches of a protein, around 250 residues, and we bind them together. We use 3 tools, and we made sure that these oligos are not present in the original data sets. We hope that it is not in the data set of AlphaFold, because the structures were new and released only in 2025. So there is a near zero chance that they are in these data sets. And we run simulations with the 3 tools, aiming to compare their performance and stability. By the way, here, instead of AlphaFold 3, we used AlphaFold Multimer. It's AlphaFold 2 Multimer. As metrics for predictions, we used DocU, MSMD, IPTM, like PLDT, Jacquard, Precision Recall. They measure global structure, like the quality of global structure, IPTM, TM, interface geometry, DocU, interchain, IDDT, MSMD, like how far the amino acids go from original crystal structures, how well the original interfaces are preserved, and then interchain contacts. Jacquard index, it's intersection over union, number between 0 and 1. Precision and recall, you probably know them. Again, 0, 1, the more the better. And here are the results. We have, remember 10 oligos. Those are averaged numbers. You will ask, what are the oligos? Here is a list of oligos. Yeah, you can find these numbers in UniProt and PDB, Protein Data Bank. And here are the metrics. So this is average over 10 complexes, mean plus standard deviation. What we look for? We look for the smallest RMSMD and the largest other measurements. First and second, we're looking for stable measurements. So if they give some error, this error is more or less around on the same level, yeah, and it doesn't jump like here, over the, it's almost a magnitude in both sides. However, we try to establish a better model for each metric, and we see that oftentimes it is AlphaFold Multimer, although it's quite old model, it's like 2023 or something like that. Yeah, but still it's the same or better, and OpenFold goes hand in hand with this model, right? So yeah, it's actually AlphaFold dominates. And here we took a threshold. For example, how many predictions for DocU are larger than 0.8. And here you want to see the larger number, the better. It means the number of high quality predictions over the threshold, right? And the winner here, again, OpenFold for DocU, for RMSD, they are equal. For IPTM, AlphaFold Multimer was the best, right? And let's jump to conclusions. So all of the models show high structural accuracy. All of them have some problems with interface geometry and recovering native interchain contacts. But that's understandable because the crystals, like the nature of the UniProt and the protein data bank, they contain a lot of mutations for the same protein. There are a lot of mutations, and there are many crystals with slightly different coordinates. So if you train AI on biased data, you will inherit this bias, and this is what we see from the results. So the interface is a little wacky, but still much better than anything we had before. And but sometimes they not only miss a large number of true contacts, but also predict many false positives. We found bolts to predict the vast majority of false positives. So it is over optimistic. I told you a little bit about this before. Scores overall for AlphaFold, Multimer, and OpenFold 3 were better and less variable, indicating their consistency. Bolt's 2 variability, high variability might be expected. Typically, usually it's a sign of overfitting. When you have jumping predictions, it means that model slightly overfitted, and it's no wonder because it was trained on affinity data, which actually very noisy. The public affinity data is very noisy. Bolts 2, like the strong side of the Bolts 2, it has a much lower level of clashes. Only clashes in when like chains intersect. In the areas they shouldn't intersect at all. And they had only 10% with one clash per prediction. And surprisingly, OpenFold had 0 clashes, while half of the AlphaFold Multimer predictions had 1 to 13 clashes. Take into account that the sample size is quite small, only 10 proteins, but yeah, it may be indication of some larger insights. In some cases, Bolts 2 had higher accuracy than compared models, but this is offset by a higher level of low-quality predictions. Also, like to summarize, to be honest, the AlphaFold is the father of the all, right? Because of the much larger data set and just origin of architectures. OpenFold is a nice open source architecture because it's lightweight, it's faster. Yeah, you can download the weights and control it completely. And bolts, it just have slightly different purpose. Yeah, it's for affinity, right? It's for structural affinity. So they are like all good in what they do. But if I were used to something, I would use AlphaFold, of course. Here we see examples of, so we overlaid a structure in gray, is the original crystal structure of a protein. And AlphaFold Multimer prediction is in cyan, Bolts 2 prediction is in blue, and OpenFold 3 is in green. As you see, they quite well and they go quite tight, and they well described major knots of the structure, like beta folds and alpha helixes. They align well here, and they misalign with the original crystal structure in loose ends. See these ends here and here. And that's quite understandable, because if you look even in crystal structures, you will see the same protein with this end dancing here and there, because it's like, you know, insanely hard to fix it in the same position in the crystal. So that's fair enough. I didn't see nothing unusual here. Here are more charts for curious viewers, how we calculated all those values. Yep, you have bars with whiskers. Every dot is every protein. Here you can see names of our proteins, 5NX3 and others. Each one have a different color. And what we are looking here for, we're looking for more dense, more concentrated predictions. The more concentrated they, it means the more stable the model. The more scarce they are, the less stable the model, right? So here we have DocU, IRMSD, IPTM, Jacquard, and other metrics. Just by brief overview, OpenFold 3 and AlphaFold has more dense bars with whiskers, and Boltz 2 is more scattered, which we already talked about in our conclusions section. Now, if you are in the lab, if you are like VP of protein engineering or VP of AI and you got a task to, or your CTO or CIO, and you just got a task from CEO that we need to run AI in all our processes, which we sometimes meet in practice. Here is what you should do. First, you must run an assessment and define bottlenecks in your pipeline. What actually prevents you from moving fast? What is the most limiting stage of your pipeline? It could be costly experimental steps like protein expression screening, right? You have too many proteins to screen and it goes too long. Or you have heat validation in drug discovery, right? You ran one round, second round. Now you want to run third round and it's a nightmare, right? So how do you deal with that? Once you identify the bottleneck, we can think now to run AI there to actually fix it. For heat validation, AI usually just narrow down the number of hits. So instead of 100,000, you will have 10,000 or 1,000. Instead of screening like thousands of proteins, you will screen like dozens, maybe hundreds of proteins. And it will speed up these steps and also will de-risk them. And then you scale it. So you do it manually, you integrate manually, you write code in Jupyter notebooks or like whatever. Then you see, okay, it works. And then you automate it. You build web interfaces, you build ETLs, data pipelines, you integrate it in day-to-day operations. And then these steps, they become faster forever. Here is some our hands-on collaborations where we saw improvements from AI. First one was HTG molecular diagnostics. We developed a multi-omics AI platform for generating drug candidates with a specific physical, chemical, and biological properties, such as log P, PK, log S, blood barrier permeability, binding affinity, metabolic stability, predicted expected metabolites, their half-time, toxicity, side effects, and actually all other things that medchems want to see. We built the platform in 4 months, and we got 23 validated candidates with confirmed biological activity. We generated probably tens and hundreds of thousands. Then we screened, ranked, and selected the top ones. Of course, after that, we consulted with, we showed them to medical chemists. They selected what they liked, and then we synthesized it and tested in the lab. And 23 of them showed IC50 nanomolar levels and a much lower than the baseline. It was a great result, patented. Now, yes, so I can talk about it. And actually, we discarded probably 90% of unnecessary synthesis. You can calculate it in terms of money. That could be millions. Think of millions in early drug discovery savings. Vanda Pharmaceutics, we built a genome analysis platform for identification of genome mutations and epigenetic mutations that led to susceptibility to COVID. And it was a collaboration with scientists and institute that did screenings and reads of whole genome sequences. We completely automated the workflows, and we screened more than 1,000 whole genome sequences obtained by high throughput sequencing. And we achieved 6 times faster data processing and actually found the genes that impact susceptibility to COVID, and also found its like genome-driven origin. The less relevant. These probably are the most insightful. So I want to close this webinar with one idea, is that protein engineering is looking more and more promising. It shows impact, and investors, both the market and scientists, see it. AI give us a chance to move from a hypothesis to results like 3-4 times faster than earlier. I will be glad to answer your questions in the follow-ups. Also, I will share my contacts in the additions to the webinar. Thank you for coming, and feel free to reach us out for advisory or helping with your pipelines, with integration of AI tools, and of course, protein engineering. Thank you and goodbye.